Bonjour à tous, merci d'être venus. Aujourd'hui, c'est Laureline qui va nous présenter ce qu'elle ne nous avait pas présenté pendant sa soutenance de thèse. Donc voilà, je te laisse te présenter.
Alright. So good morning, everyone. So the presentation will be in English. I'm Laureline Dallo. I defended my thesis 3-4 weeks ago. And today I will present how nitrogen fixation influence high-trophic level response under climate change conditions. So it's something I didn't present during my thesis defense. It's a new topic. The only thing that will be the same is the model presentation. Okay, so let's dive to the presentation. Just a little bit of context from CEMIP 5 to CEMIP 6, CEMIP 1 to CEMIP 5, we observed a decrease in primary production under climate change because when there is an increase in stratification, we usually observe a decrease in primary production. But it's a little bit different between CEMIP 5 and CEMIP 6. So between the two versions, we have a similar sea surface temperature over time between the two versions, but we have an opposite response in primary production. With the CEMIP 6 that exhibits an increase in net primary production over time under climate change. And this is due to the new nitrate fixation parametrisation, PISCES V2. So this had an impact on the marine ecosystem model, where we had a biomass that was with a lower decrease than expected. So we tried to fix this. Well, biogeochemists tried to fix this, and they did a new parametrization, which is called PISCES V2 fix. With this version, this is the one I use for my thesis defense, but I wanted to see what is the difference in high-trophic level responses between the two versions, in PISCES V2 and PISCES V2 fix. So I will quickly present the two versions of PISCES. We have PISCES V2 parametrization that induced an unresolved source of inorganic phosphorus derived from unstable dissolved organic phosphorus and independent of the rate of nitrate fixation. Where's my mouse? Okay. PISCES V2 fix, on the other side, assume that nitrate fixation is limited by the availability of dissolved organic phosphorus and that the organic matter produced by diazotrophy is enriched in nitrogen relative to phosphorus. In PISCES V2 fix, explicit diazotrophy transfer nitrogen and phosphorus to dissolved organic matter. And this version also considers different temperature sensitivity in nitrate fixation. So just to simplify, we will call the old version H-fix for high nitrogen fixation and the new version L-fix for low nitrogen fixation. So to observe the impacts on the higher trophic level, we're using the APECOSM model. So APECOSM model has inputs from PISCES, which give us the light, the plankton, and the dioxygen. And it also uses NEMO for the ocean physics that give us the current, the temperature, the 3 dimensions, and a 1 degree grid. They interact with each other and enable us in APECOSM to simulate 6 high trophic level communities. So we have the small coastal pelagic, small and medium pelagic, and tropical tuna that live permanently near the surface. We then have the mesopelagic migrants that live at depth but feed at night near the surface. And we have the mesopelagic feeding tuna that live near the surface but feed during the day at depth. And just to finish, we have the mesopelagic resident that inhabit permanently at depth. So to simulate these communities, APECOSM uses a mechanistic approach with fundamental theories and processes, such as the dynamic energy budget, DEB. It is also a trade-based approach model. It considers the phenomena of schooling, the passive transport by currents, the active movement of organisms, and it also considers ecological processes such as size, structure, trophic interaction. So the goal is to model low-level processes and see how the mechanism and structure of ecosystems emerge from these lows. Now that we have the models, let's have a look to the simulation. So we will compare here the response of L-fix, the new version, with H-fix, the old version. So when we're looking at the nitrogen fixation, we have an increase for both simulation, but for the old version, it's 4 times higher than the new one. About the nitrate concentration, we observe for both a decrease, but less than 1% for the old version, H-fix, and around 12% of decrease for the new version, L-fix. So, as expected, the night primary production has returned to a decrease under climate change condition with minus 11%. And plankton, like phytoplankton and zooplankton, also exhibit a decrease in both simulations, but we have a difference from 2 to 3 times more decrease with the new version. So, and between also phytoplankton and zooplankton. So we have a trophic amplification that we can also observe in higher trophic communities with minus 24% under the new simulation. But we also have high heterogeneity between the communities, with some communities that are not so influenced by the change of nitrogen fixation, like the small coastal pelagic and mesopelagic feeding tuna. But we have other communities like tropical tuna and mesopelagic migrant that exhibit a high difference between the two simulations. So this pronounced heterogeneity in high trophic level responses cannot be explained only by a low trophic level because there's so many differences between the communities. So we'll go back to the nitrogen fixation and try to understand how it's influenced the higher trophic levels. So here what you can see is the anomaly between H-fix and L-fix at the end of the century. So in red, it's all the region where there is more increase in nitrogen fixation under H-fix. When we compare with the high trophic level anomaly, so you see here the hatches that represent what you can see on the left. It's the H-fix concentration. And we can see that the hatches regions do not match the color that you see behind, which are the high trophic level anomalies. So there's something we cannot explain now that makes that it's not the same results. So when we look at the nitrate concentration anomaly, we have kind of similar pattern with the N-fixation regions. But we have, for example, here near the Humboldt and the Koltong, red patches that you cannot see on the H-fix results. And this is due to the currents. So with the wind, we have surface secret ocean currents, and it is then exported due to the Earth's rotation northward and southward, and then go deeper to the ocean. So this movement, we can see here on this part of the transect that is represented below. And you see that the anomaly go deeper in the ocean before going up again. And this rising in the currents, we can see here on the other transects that you can see here is represented on the lower right. And you see that you have the nitrogen fixation, the nitrate that go up with the upwelling. And so in this region, we have an increase in phytoplankton and zooplankton. Here you can see the anomaly map with the clear movement of the currents and the patterns. But again, so we have the anomaly in zooplankton and we have the anomaly on some of the communities. And we can see that it's still not similar. So we'll try to understand why the zooplankton do not drive the response of higher trophic level communities. I will not look at all the regions, but for example, we start with the Kuroshio current, which exhibit a similar response. We'll then see other regions like the Gulf Stream where depending on the communities, we have similar responses, but not for all the communities. And then we will see at the equatorial Pacific, where we have clear positive response in zooplankton, but negative response in the higher-trophic-level communities, especially in the west and the central part of the ocean. So let's dive to the Kuroshio currents here. So here is represented the transect that you can see on the upper right. And it's here the pattern of the epipelagic community, so small and medium epipelagic community and tropical tuna. So here, when you look at the zooplankton, we see a strong positive anomaly in the west parts, which decrease a lot, but stay positive on the western part. And this drive bottom-up control. So we have positive anomaly on the higher trophic levels, except in some size classes. So here, just to explain, you have the epipelagic community, small and medium epipelagic, and tropical tuna, all along the transect. And here on the y-axis is the size classes. So large organism on the top and small organism on the bottom. So except for some size classes, we generally have a positive response all along the transect. And this is due to the zooplankton positive anomaly. So you have a bottom up. But as we have more high trophic level organism, we also observe a top-down effect with larger organisms that exert a predation pressure on smaller organisms. But you can see here with the stripes, where we have large organisms that feed on smaller organisms that release pressure on intermediate organisms that increase pressure on very small organisms. So we have in this region the multiple effects of a bottom-up and top-down control. Now let's have a look to the Gulf Stream currents. Here we have a positive anomaly of zooplankton, but also negative anomalies. Again, here is the transect that we are looking at. I just selected two boxes, one the red with positive response and another a blue one, just to see what size classes are driving the response that you can see on the map, the more numerous organisms. So here what you can see is that for epipelagic communities and mesopelagic migrants, we have the response is driven by the size classes lower than 6 centimeters. And looking at the transect, so again, we have all along the transect here with the size classes on the y-axis, you can see that in the red box, which correspond to this zone, we have a positive response of smaller organisms and a negative response in the zone that corresponds to the blue one. So what you can see is that when there is a strong positive anomaly in zooplankton, we have this bottom-up effect with all the organisms that increase. But then there is a decrease due to negative anomaly of zooplankton or too small positive anomaly to enable the organisms to sustain. And what we can see also is that we have like a propagation of the positive anomaly on the larger size classes that are affected by the currents. So here you have the effects of the bottom-up control, the top-down control, and the currents that affect the response of high trophic level organisms. We have the mesopelagic migrant that have a different response because they are not, they do not have the same, the current do not have the same effect on them as when they are near the surface. They are submitted to the current or go eastward, but when they go deeper during the day, they have the current that go on the other side. So they maintain mainly near the coast where they exhibit a strong positive anomaly. Now let's look at the equatorial Pacific. So here you can see the transect. Again, I choose two boxes, one with positive anomaly and negative anomaly to see which size classes drive the pattern, which you can see on the map. So in this region, we have zooplankton positive anomaly everywhere, but with a peak at the extremities. And about the organism that drive the response, it's organisms that are lower than 3 centimeters, even smaller for the mesopelagic migrants in the blue boxes. So you have here the response of epipelagic communities. What you can see is that they are affected by the positive bottom-up control on the eastern part near the Humboldt. That propagates westward by the currents. But as there is less less zooplankton eastward, the small organisms are not able to sustain anymore, so only large organism propagates. And we have a negative response on the west parts. On the other side, we have the mesopelagic migrants that are less affected by the currents. And so they maintain longer on the western part, on the eastern part. They also have a negative effect when there is less zooplankton, and they have then a positive effect. Just to explain, here we have the negative response because in the tropical equatorial Pacific, the water is warmer, so the organism, the metabolism increase, so they grow faster, they have a shorter lifespan and need more food intake to sustain themselves. So that's why you have a negative response here, because the lifespan is shorter. But what we're wondering is why there is a negative response in the western part, where there is a peak in zooplankton, positive anomaly in zooplankton in this region. And this we can explain when we're looking at the ratio between mesopelagic migrants and epipelagic communities at the end of the century. So here you can see for both simulation H-fix and L-fix, the ratio of migrants compared to epipelagic biomass at the end of the century. And the dotted lines are without climate change. So the ratio changed a lot. And it's because there is a decrease so strong in epipelagic communities. You can see here the zoom of the size spectra in the region A. So in blue, you have the epipelagic communities. So you see a strong decrease between the dots and the solid lines. While in mesopelagic migrants, decrease is not as strong. And in log-log scale, we even don't see the difference. So at the end of the century, there is relatively more mesopelagic migrant than mesopelagic communities in this region. So mesopelagic migrants benefit from the top-down control by zooplankton, but they are very numerous, more numerous than epipelagic communities that exert a predation pressure so high that epipelagic communities have a negative response. So the take-home message of this presentation, just do a quick resume of all of this. We have the nitrogen fixation and remineralization that occur in the ocean and is then transported by subcurrents, where we have more zooplankton that exerts a bottom-up control on higher organisms. Due to the currents, the zooplankton anomaly is exported, and the bottom-up control is then exported. But we also have a top-down control due to higher biomass in high-trophic-level organisms. On the Gulf Stream, we have the same effect, a bottom-up control that drives a top-down control. But as zooplankton go to the negative anomaly, we have a negative bottom-up control with a negative response on high trophic levels. And with the currents, we have the top-down control that propagates all along the currents. In the equatorial Pacific, we have the bottom-up control, the top-down control, a negative response in the bottom-up control, and on the western parts, we have a bottom-up control that benefits mainly on the mesopelagic migrants because they are more numerous than the epipelagic communities, and exert a top-down control on smaller organisms and pelagic communities. So this presentation tells that change in nitrogen parametrization has a significant impact in trophic chain under climate change, and it highlights the importance of accurate nitrogen fixation parametrization for understanding the future of marine ecosystems. Thank you for your attention.
Thank you very much, Laurelie. Is there any question? Here?
Okay, go. Okay, so I have lots of questions that are very interesting.
Je pense qu'on va pas t'entendre.
So I have all sorts of questions, but I'm not going to ask them all. So my first question, okay, so this one change in the parameterization of CMIP seems to make a huge change in predictions. It seems I was unaware of the scale of this change. If I was a climate skeptic looking at this, I would say those scientists, they just push buttons, and if something comes out and you can't believe any of it, right? Because you have a trend that's going up and then it's going down, or vice versa. So can you like explain what is changing that's making that drastic shift? And is 6 better than 5, or is it just different? Like which are we to believe more?
I think it depends on everyone. I would say that for me, CMIP 6 is more relevant. The new version, which will be CMIP 7, is more relevant because we have again the decrease in primary production, which is, well, we don't know in the future, but it seems more logical to have a reduce in primary production under climate change because of the increased stratification, increased temperature of the water. So I would say it's more relevant, but again, everyone can say we don't know, it's the future. So
what has changed? Like what levers have they moved to make those changes happen? Maybe I just didn't understand it when you explained it.
Here you have all the differences between the two versions. Sorry, I'm not an expert in biogeochemistry, but it's just in PISCES V2, there's not a lot of link between dissolved organic phosphorus and the nitrate fixation. On the new version, you have a link. You also have an effect on the particulate organic matter. And there is also, I think, what makes the huge difference is the difference in how they calculate the influence of the temperature. But I don't know how they calculate it. But what we can see is that even if you have a huge change, for example, in nitrate fixation and an opposite response in net primary production, we still have the same tendency, well, a negative response in phytoplankton, zooplankton, and higher trophic level. So it does not change the trend. It's changed the amplitude on higher on phytoplankton, zooplankton, and higher trophic level organisms. So there's uncertainties and we have to show them, but at least the tendency is still the same.
Je veux bien reposer la question encore plus néophyte. La nouvelle version, est-ce qu'elle intègre des processus connus qui n'étaient pas intégrés dans la version précédente? Ou est-ce que ce sont des nouveaux processus de minéralisation qui ont été compris depuis l'ancienne version?
I don't know. Je sais pas.
Et la deuxième question qui allait avec ça, c'est de combien on est loin de la connaissance des processus de minéralisation, de la connaissance actuelle dans les PISCES, dans les dernières versions de PISCES. Est-ce qu'on voit l'impact, l'effet levier que ça peut avoir sur certaines parties de l'écosystème?
Je ne sais pas si c'est des nouvelles connaissances ou aussi il y a eu un objectif de simplification et finalement on est revenu à un peu plus de complexité parce que les réponses étaient complètement différentes. Et la deuxième partie de ta question,
Et avec cette nouvelle version de PISCES, V2 fixe,
oui.
est-ce qu'on a encore tout un tas de processus, des éléments de la minéralisation qu'on connaît mais qui ne sont pas pris en charge par le modèle? Ou est-ce que là, on est allé maximum, entre guillemets, de la complexité possible vis-à-vis de la connaissance disponible? C'est ma question.
Vis-à-vis de la connaissance disponible, je ne saurais pas trop dire, mais c'est vrai que même maintenant, il y a encore beaucoup d'incertitudes, autant d'ailleurs pour la fixation d'azote que par exemple aussi pour la manière dont on représente le fer aussi, par exemple.
Mhm.
C'est vrai que la biogéochimie a beaucoup d'incertitudes. C'est assez difficile à comprendre.
C'était la question d'un béotien.
Pour aller dans le même sens, bon, bravo, c'est très intéressant, très élégant, c'est très intéressant de voir ces changements de bottom-up selon le spectre de taille. Mais compte tenu de ce que tu montres, moi, l'envie criante que j'ai, c'est d'appliquer ça non pas dans le futur, mais dans le présent, et de tester, parce que c'est très intéressant. Alors, on voit que les processus sont très différents. Et moi, j'ai qu'une seule envie, c'est de voir une application dans le présent, pour voir ce que ça change dans le présent, et d'utiliser des zones data rich pour essayer de voir si ce qu'on retrouve réellement avec des données, c'est de configuration du modèle.
Bah, dans le présent, ça ne change pas trop parce qu'en fait, la façon dont ça a été paramétrisé, ils l'ont fait sur le présent. Donc, ils ont les mêmes valeurs.
Changeons les paramètres.
Il y a juste la fixation d'azote qui a changé.
Oui, mais ça ne change rien dans le présent.
Et maintenant,
Si
tout va
tu mets
bien.
les deux versions de PISCES, ça ne change rien dans le présent.
Non, justement, en fait, c'est pour ça qu'ils l'ont vu que quand ils ont fait les projections climatiques.
Donc, d'avoir la version très simple ou la version beaucoup plus avancée, ça ne change pas.
Non, ou alors c'est négligeable.
Ils sont calibrés sur le présent.
C'est
Donc,
calibré sur le présent.
Je pensais que malgré tout, vu la différence et dans des processus biogéochimiques entre l'ancienne version et la nouvelle, qui sont quand même très fort, j'aurais pu penser que même dans le présent, il y a des différences. Il n'y a pas de différence.
c'est scientifique qui peut faire marcher tout calibré sur le présent.
Il y a peut-être des petits changements, mais c'est pas, enfin, c'est négligeable. Et en fait, les divergences apparaissent vraiment avec le changement climatique, les scénarios climatiques.
D'autres cas. Vas-y.
Justement, enfin, il y a quand même eu un changement climatique qui a commencé, mais du coup, si tu calibres sur jusqu'à il y a 10 ans, et tu t'évalues sur les 10 dernières années, est-ce que ça a été fait ou pas du coup? Quelque chose comme ça, quoi.
Biogeochimiste. Euh, Moi, j'ai pas vu d'énormes résultats. Après, j'ai pas comparé par exemple, 2010 et 2010-2020. Après, il y a aussi, c'est difficile parce qu'il y a aussi d'autres choses qui peuvent induire une différence de réponse. Donc, moi, je l'ai pas fait. En tout cas, vraiment, ses parents, c'est des périodes de 10 ans. Mais même en général, j'ai pas vu beaucoup de différences en regardant sur l'historique présent des deux versions.
Une question à Montpellier, apparemment.
Il y a un commentaire
Et
d'Olivier.
il y a
Thank you, Lorlin, for the presentation. I was wondering, so this change in the hypothesis regarding fixation, then it affects not only your model, but all the models of climate projections. Are you aware of other studies that also looked at the robustness or the effect of this change, maybe in trophic levels or in other aspects of the projections, and if their results are similar to you, so that it can change a lot what's happening, but the trends are still the same, or, well, are you aware of other works that looked at this new difference and what it implies?
On a higher trophic level, no, I'm not aware of other studies like this.
Actually, you have maybe the fish meat paper that compared the global simulations between CEMIP 5 and CEMIP 6. And basically, they did exactly the same kind of experiments than Laureline. They compared HTL projections between CEMIP 5 and CEMIP 6, including CEMIP 6 with high nitrogen fixation. And basically, you have the differences in biomass simulation in
right.
both experiments.
Yes, but I mean just in a total animal biomass.
Yeah.
So we do not see the top-down effect and all the
Okay, specifically.
different effects that are presented.
Okay, you went deeper in the analysis. Okay, I see. Well, thank you.
Quoi le commentaire d'Olivier?
Le chat.
Olivier peut prendre la parole.
Alors, Olivier, la conséquence.
Et la réponse.
Je peux le faire, je peux le dire à l'oral, ce sera plus facile. The main difference between both version of PISCES actually is the response to temperature of nitrogen-fixation. Namely, in the high fix version, the nitrogen fixation accelerates much more with temperature than it does with the low fix version. And you know, when CEMIP 6 results appeared, so the last round of simulation of the IPCC, then we had most biogeochemical model were predicting a decrease in net primary production, except PISCES because of this. So that was a problem. So we looked at that, and Olivier Aumont and Laurent Bopp looked at that in details, and they found out that it was mostly this response to temperature that was driving this pattern, and this response is not well known. We don't really know, you know, how nitrogen fixation will actually respond to temperature in the future. So it can be seen as a sort of sensitivity experiment that we are doing here to a process that is not well known, and it's quite interesting. And to extent what Lorena said, I think it is important to stress that the results are robust. I mean, the in any case, we predict that climate change will lead to a strong decrease in high-trophic-level. And the differences are second order. They concern different communities, the magnitude, and also what's maybe Laureline hasn't shown very much is that it is actually anomalies of anomalies. Uh, so the projections are robust in their response, but there is a complexity that propagates these uncertainties through the ecosystem, and that can have regional and remote effects that is important to take into account. So that was my comment.
Just ask a question a bit in response to that, Olivier. So thanks. That was very interesting. The one thing about this change in how nitrogen fixation works as a function of temperature is that it's not just a degree change, it's a conceptual change, right? One of them is exponential and the other is dome-shaped. Is there like some sort of theoretical, I mean, I don't even begin to understand exactly how nitrogen fixation works, but is there some sort of theoretical reason why we would expect a dome-shaped response instead of a like monotonically increasing response?
Laureline, you want to try to respond or?
The response.
Okay, my take on this is that all the biological responses to temperature are dome-shaped. It's a rule because, you know, at low temperature, you know, the enzymes lose their ability to work normally, and at high temperature they denaturate. They lose their quaternary structure and they lose their ability of working. So the response to temperatures is always dome-shaped. But I think Olivier did that because he had no observations, no experiments to constrain this in the model. So he took just an Arrhenius function, which is exponential, disregarding the limitations, and actually it had important impacts. And since you have to keep in mind that there is a very strong agenda with IPCC, and we have to submit the simulations at very strict dates. So when they submitted this simulation, they actually didn't see what was happening. They produced the simulation. The model was tuned on the historical. It was working. You can't distinguish both versions on the historical because they are tuned to work on the historical situation. But the divers in the project and they didn't see that before submitting the simulations to the semi. And the thing is that we know that this response to temperature is dome-shaped, but we don't know when it decreases. So we need more experiments and we need more, I don't know, but we need to learn more on the processes of nitrogen fixation response to temperature. That for sure, it's an important uncertainty. But there are many others like this, and this propagates through the models, of course, and that's what Laureline is pointing.
Other questions?
Possible, yes.
Oui.
Olivier mentioned it's anomalies of anomalies, and I had a question on that. Can you go on to the pure ratio for outputs, for instance, just to make sure I understand? Yes. So it's a so you are looking at the difference between the two versions, H and L, or the new version of the model. And what is delta H and delta I? It's the difference between what and what?
Climate change and no climate change difference with climate change conditions.
In which and a given scenario?
Yes, high emission scenario.
Okay.
Sorry, I didn't explain that.
And if we look at the right maps, outputs for the epipelagic and mesopelagic segment.
To interpret the...
No, but you have 80% of difference in some areas, 80% of positive or negative difference between the two versions.
80%. Yes. Yes, depending on
Nearly
classes.
simple to double. Why the difference in terms of the primary production or zooplankton production is only 20? Is it?
Yes.
Okay.
Right. No, monopolize that. One question that I had that I also had when you were presenting your thesis was that oftentimes in your models, you do have this like spatial, the spatial separation between sort of the primary process and the secondary process that you attribute to currents and transport and things like that. I'm wondering, particularly when you get to higher trophic levels, isn't like the capacity of those species to migrate a really important factor? And are you sure like the migration rates are important, are correctly parameterized, things like that? Do you get what I'm saying? Am I making any sense? No.
I mean the swimming speed and the
Yeah.
is it correct?
Yeah, like because you do have like a lot of spatial separations, it seems, between, say, the upwelling and where you get the primary production, and then where you get the increased zooplankton and further trophic levels. I would imagine those things are really sensitive to how you parameterize movement.
Yes, it is. Well, I mean, when I compare with observation, I also see this for example, the tuna fisheries. We also have in the equatorial Pacific this kind of pattern where there is more fisheries northward and southward, and not exactly at the equator.
Yeah.
So there is clearly an effect of the currents. Is it well parameterized? Well, I think there is always adjustment to do, but for now, at least for pelagic communities, when I compare the observation, I see same patterns. Similar product. And for the swimming speed, the active movements, so we used the swimming speed of tuna as a reference. And as we do now for all the communities, we applied for each community the same.
And Olivier, it's mostly the effects of currents that we see here, and it's quite well told.
Yeah.
Other questions online? See? Bon, eh ben, très bien. Eh ben, merci beaucoup.
Merci Laureline.
Merci d'être venu. Merci beaucoup pour ta présentation. Jeudi prochain. La nouvelle vacances.
Pourquoi il dit qu'il est 7 heures du matin?
C'est elle qui devrait dire. Non, tiens, on va.